Okay, can everybody hear me all right? Thanks for the invitation for, as Noel calls it, a return performance. And I thank all of you who are managing to stay to the very end. What I'd like to do for you today is just tell a story or weave a story a little bit about how my interest in tolerance has led to progression through interest in applications in autoimmunity and in hemophilia, which, while it's not an autoimmune disease, provides a very nice model system. So this, by the way, is where I now work, which fortunately for me is by talking a little bit about Factor VIII and hemophilia. And don't everybody get up and leave because you're not interested in hemophilia, but it's actually a very fascinating disease and important. And then I'm going to talk very briefly about the historical work we've done with using gene therapy to induce immunologic tolerance and how that has led us to new insight. I'll talk about some of the mechanisms we know and how this works, and show a movie of some of the interactions that are going on during tolerance induction using this method. Then finally finish with where this has led us to understanding of some epitopes that may play an important role in tolerance induction and potential applications in autoimmunity and in designing biotherapeutics and overcoming some of the problems of biotherapeutics, including some of the side effects of a number of the drugs that are being used to treat autoimmune diseases, etc. So obviously, for this audience, I don't have to tell you that immunogenicity is controlled by a number of different factors. But that's important in the pharmaceutical industry as well, because so many therapeutics, even humanized therapeutics, humanized antibodies, can be immunogenic. And without going through the list of all these different factors, including clearly the pathology and the inflammation going on, there are a number of properties that need to be considered. And what I want to talk about is the biologic properties, because the antigens that are targets in autoimmune diseases, or in the case of hemophilia, are not simply proteins with a conformation with epitopes, as an immunologist tends to look at them with those blinders, but rather they have important biologic properties. So let me briefly introduce hemophilia for those who aren't that familiar with it. So patients with hemophilia A have mutations in Factor VIII, a variety of different mutations which lead to a defect in their clotting and procoagulant function. The interesting thing is, one of the problems is you can treat these patients with Factor VIII, but up to about 30% of them, depending on the nature of the mutation, make an immune response to Factor VIII. And you can even make an immune response to Factor VIII in normal mice that have their own Factor VIII, and in rhesus monkeys and cynomolgus monkeys that we've done in collaboration with people at NIH and at University of Maryland. And that's when it's given intravenously, which, as all immunologists know, you never immunize an animal with a protein intravenously. It induces tolerance. Factor VIII is immunogenic. So really, what is really the secret? Well, there's my punchline. Thrombin seems to be one of the secrets. One of the downstream products of Factor VIII and the procoagulant cascade is thrombin activation, cleavage of fibrinogen to fibrin, platelets, and then platelets. Then once they aggregate, they release a whole bunch of immunologically active substances. So if you have any interest in a new field, as I said in The Graduate, the magic word for those who are old enough to remember, plastics, I'm going to say platelets. Pay attention to platelets. All right, so here's an example of some data. I'll do this very quickly. You immunize with Factor VIII intravenously, you make a nice immune response. Notice this is a log phase. You immunize with OVA, you make a mediocre response. If you give Factor VIII plus OVA, now you get a good anti-OVA response compared to OVA alone. If you block the clotting activity of Factor VIII with these leach product Hiridin, which is a thrombin inhibitor, you inhibit the activity of Factor VIII, the immunologic activity of Factor VIII, and if you actually give thrombin plus OVA, now you get immunogenicity of OVA and thrombin acting as an adjuvant. Now, I'm not suggesting that we use Factor VIII as an adjuvant. I'm not suggesting we use thrombin as an adjuvant, but I want you just to think about this in the context of the biological activity of the proteins we use, the proteases, the enzymes that we use in studying the immune response, and some of the targets that we've heard about already today that are important antigens in autoimmunity. So I'll leave that story out as the first introductory story, but just file it away as something I think is important. So over the years, our laboratory has used gene therapy to induce tolerance, and not necessarily to express genes to replace a gene, but rather as a mechanism to induce tolerance. And we've used B cells with immunoglobulin infusion proteins as the vehicle, as the vectors involved in this. And the key strategies was based on very old work based on the fact that immunoglobulins can be very tolerogenic when given at certain doses, and then we decided, and this was started by Elias Zambides, who many of the people here at Hopkins know. He was an MD PhD student with me in Rochester before we moved here. He engineered target antigens at the N-terminus of an immunoglobulin heavy chain. We then put this into lymphocytes and actually myeloma cells, trying to make fusion proteins that eventually put it into B cells to get the host to continue to make these fusion proteins that would be tolerogenic, based again on the tolerogenicity of immunoglobulins that had been done by Burrell over 30 years ago. And it turns out, obviously, the critical thing is these get processed. The peptides are presented by the B cells in a certain context, and then they induce tolerance. And the B cell selects the relevant peptides, which is kind of nice because you don't have to know the exact peptide that's the target, just an antigen. Now, in hemophilia, it's very easy. We know what the antigen is. It's Factor VIII. In MS or diabetes, there are a variety of antigens, as David has talked about earlier today. So this is just a schematic of the procedure. It's simply a matter of creating a retroviral construct and putting it into B cells that have been activated, so they will now accept this construct, and then we just put that into naive, ablated mice. And in fact, we also put it into immunized mice, because that's the more clinically relevant situation. This is a slide I've shown many times. The purple or magenta is to represent the IG heavy chain, and we've inserted a variety of antigens ourselves or our collaborators, initially working with IRBP with Rachel Caspi at the Eye Institute in experimental uveitis, but we've used a number of antigens in EAE models, a number of antigens in diabetes, and with Kamal Magdo at the University of Maryland, his group did it with a bovine heat shock protein that was important in adjuvant arthritis. With Factor VIII, instead of making the full-length Factor VIII, we simply took two domains of Factor VIII that were important targets for the inhibitory antibodies. I mentioned again that a number of patients make antibodies to Factor VIII, which essentially render this therapeutic ineffective. So we wanted to try to get rid of those antibodies and the T cells that are turning on those antibodies in this response. And I only put this in here in case anybody is interested in gene therapy in general. Think about what kind of genes you're targeting, whether you're looking at Pompe's disease or something like that. This is a very nice method in which you can do that. I'm happy to collaborate and send out the constructs for anyone who wants to do it. Okay. I don't want to spend any more time on this, but this is a list of all the publications that have been done in the models that I just showed you in that cartoon slide. Basically, it works, and it works in naive as well as primed animals. I'll just show you one bit of data with hemophilia before I move on into what I think is the more exciting and more interesting and more recent data. Now, these are activated with LPS. I'll return briefly to the way we activate them. Then those cells are transduced with C2 and A2 Ig vectors and put into naive or immunized mice, and then we follow the immune response. And we try to do as many different parameters of the immune response as Polly Matzinger would want us to do, because you have to measure everything. But one of the important things we measure is the inactivation of Factor VIII in terms of Bethesda units, which is the gold standard. That tells you that you're inhibiting the immune response. A Bethesda unit is simply a measure of the inactivation of Factor VIII. That's Tai Chi. You know, I should point out, a lot of people think a Bethesda unit is something one might aspire to live in. This is actually a quote from Mark Jenkins. It's an old joke, but I love it. Okay. And this is how well it works. This is the control using actually a soluble antigen from the retina as a control construct. Here C2 plus A2 showing a maximum effect on the anti-factor VIII inhibitor titer. And even C2 alone or A2 alone will work to reduce the titer. And this will also work in primed mice. So it means patients who already have inhibitors, as long as you can get rid of the plasma cells, you can now prevent further inhibitor production. Okay, this summarizes about 10 years of postdoc and graduate student work, but basically what should be obvious by now, you need MHC class II on the B cells. The cells don't actually secrete this fusion protein, which was totally against our original hypothesis, but that's the way it works. You need B cells, B cell knockout mice, but bone marrow from B cell knockout mice doesn't work. The IG scaffold is important, and it led to a very interesting collaboration I'll tell you about in a few moments. It's not dependent on FC interactions or on IL-10, much to our disappointment and to the people who believe IL-10 produced by B cells is really critical. But rather, there are interactions between co-stimulatory molecules and CTLA-4 that lead to the recruitment of regulatory T cells. And then finally, activation by different toll receptors may be important. And the reason we came across this is, while we're mouse doctors, what we really wanted to do is get this to the clinic. And so we had to figure out a way to activate human B cells so we could transduce them with this retroviral vector. And so we knew that LPS would not be a good stimulator for human B cells. So we tested mouse B cells with a number of different stimuli and found that everything worked to stimulate, to transduce, and to get good expression and tolerance induction with everything except for CpG. CpG B cells just didn't work. And so at this point, we took a little detour and a distraction, but it led to an absolutely wonderful collaboration with Mike Cahalan's lab in Irvine, California. So basically, what we did is we would take B cells, activate them with either LPS or CpG. These B cells we proved, and these are actually using transgenic B cells. It worked the same way. These are tolerogenic, these are not. They would be labeled and with different dyes, and put on a plane and sent to California, because this is what we call the FedEx experiment. Keep B cells cold, they'll survive 24 hours. Jan Su did this work. Jan is still with me. Then they went to California, and Melanie Matho in Mike's lab would inject blue labeled cells that are OT2 transgenic, so that every T cell had a chance to interact with every B cell, and they'd see whether we could see a difference in the interaction of the different cells. And then they would analyze it by 2-photon microscopy. This is Mike. Ian Parker made the machine, and Melanie is the graduate student. So I want to show you a couple of movies really quickly. I hope they work. In general, we label the LPS B cells red, CpG B cells green. Sometimes we reverse it just to make sure there's no artifact. And you'll always see blue cells that are the TCR transgenic. This is going to be 2-photon microscopy in the inguinal lymph node of a living animal. This just shows if the follicles had been labeled, you'd see that they'd be over here. There are a lot of T cells in the intrafollicular space. There are a lot of LPS B cells here and some CpG B cells, and some LPS B cells. They all migrate. LPS B cells migrate a little faster and a little better, and they stay there a little longer. Basically, what we did is what I call speed dating. We actually did a movie where we looked at these cells coming and sampling each other and deciding who's going to, I hate to use this expression with a young audience, but who was going to hook up and continue the interactions that would lead to tolerance or immunity. And this shows you essentially what happened. With LPS B cells, they bind and they track together. So you see blue and green dots linking together. CpG B cells, they might hit, but then it's obviously not a very effective date, and they break up. And this is a consistent finding. This shows synapse formation with the LPS B cells and much shorter formation, less strong synapse. And in general, the LPS B cells interact much longer than the CpG B cells. So that was really fascinating. It suggested to us that there's a longer interaction necessary, but it turns out it wasn't that different from what T cells and dendritic cells interacted. So that was just part of it, and we wanted to understand what else was going on. Why were these interactions different? This is simply a summary of what was many hours and days and nights of work to analyze it. There's at least 2 to 3 fold, as I just showed you, a longer interaction for tolerance to occur as opposed to a non-tolerogenic interaction. This is actually one of the movies that was made more recently, and it's kind of hard to see this, but the green cells are actually LPS B cells. The red cells are CpG B cells. Now I stopped it here because here is a CpG B cell about to interact with an LPS B cell. A CpG B cell about to interact with a TCR transgenic T cell. And you'll be able to follow the tracks for a moment, but the important thing is it's almost like a chemokine gradient. Like the B cells know something's going on there. This is at 18 hours. They're being attracted to this party, so to speak, that's going on. So watch what happens, and it may be a little tricky, but what happens, that T cell is now with this different B cell. The LPS B cell has actually kicked it off. You can't see that very well here, so let me just go to the next slide where you don't have the cells and you just have the tracks. So here's the interaction. They're coming together, and they come together, and then, oh, boom. Well, actually, an LPS B cell got together with a T cell here, but then here he goes. They've paired off, and who knows what they're going to do the rest of the night. But that's the interaction that's going on. So what's happening is the interactions are just not as strong. We know that there are differences in integrins, etc. There are no differences in cytokines that explain this. So somehow the interactions have to be strong enough in order to get tolerance to occur. All right, I think enough on that. I just love those movies, though. So there's a swarming effect. And very briefly, we know that CTLA-4 is important, and what I just want to show you here is, if you see here, the cells are labeled different colors, but you see the red and greens get together and they hang together a long time. Here, they get together and they fall apart. And what she's done is she injected anti-CTLA-4 to break up the interaction. We don't know, and we don't have any evidence that these are actually Tregs. We actually don't think they're Tregs at this point, but we know that Tregs are important, and maybe this is helping to recruit Tregs. Okay, so in summary for that part, and I know I went rather quickly, but I wanted to emphasize the last part and bring it back to an autoimmune disease and what we think is going on. What we've learned over many years is that there's an interaction between a B cell that has to present the antigen in class 2 to the T cell receptor on the T cell. Co-stimulation, CD80 and 86 are involved in bringing them together. This may lead to an interaction with CTLA-4 at a negative signal. We know these B cells make IL-10, and they make more IL-10 when they're LPS stimulated than when they're CpG stimulated. But we don't think IL-10 is involved for a number of reasons. Basically, every experiment and every combination using IL-10 knockouts has not worked. And I know of at least 2 other laboratories that have exactly the same result in similar model systems. So IL-10 is not required to be made by the B cell. But Jansu now has shown that IL-10 probably is made by the host, because you can block IL-10 receptors in the host, and that will then block tolerance induction. The other thing that could be going on, and we actually think this is the dominant mechanism, is the CTLA-4 interaction with B7, and the CTLA-4 is coming from a FOX3 P3 positive regulatory T cell. This method does lead to an increase in regulatory cells and increased functional regulatory cells, work that Jonathan Skupsky and Yoni have done in my lab, and Jan have done in my lab. So that's the current theory in terms of the mechanism. That's what we think is going on in gene therapy. But we did all of this with immunoglobulins, and why did we use immunoglobulins? Well, a dozen years ago, we thought, well, we'll make a fusion protein and the cells will secrete it. We want to put it on an immunoglobulin scaffold, but it always worked better when we used the immunoglobulin. Even though we know the immunoglobulin is not secreted, we always got better and more long-lasting tolerance. So a few years ago, I ran into Dr. Annie de Groot, who is an infectious disease specialist, a vaccine specialist, but she runs a company that maps epitopes. And she came and said to me, I think I know why the IG works better. we've discovered that there are a number of peptides in immunoglobulins, predominantly in immunoglobulins, but also in many proteins, and that by mapping epitopes, they found a number of recurring motifs with strong signals to HLA class 2, HLA and H2. So mouse and humans share these. They bind to class 2 and apparently would presumably be able to signal to T cells because they're being presented by class 2. They are very promiscuous, meaning if you look at a whole range of different HLAs covering 90% of at least the Caucasian population in America, you will find that they will bind to most or all of these. They're in all IgG molecules. They're not in IgA or IgE or IgM. And they're in both heavy and light chain, predominantly in heavy chains in certain domains, and I'll show you in a moment. And they are spread through mouse, cat, and camel, and go figure that one, but they're obviously evolutionarily conserved. The reason she found these is companies would come to her and say, I just had blah, 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 mab, and it's immunogenic. I'm getting adverse events in my patients. Can you tell me what are the important epitopes? And they would go and try to map the epitopes, and sometimes they could change them and improve the efficacy. And sometimes they found that these epitopes with these that would match an MHC binding epitope never showed up as immunogenic, never showed up. And they said, well, maybe they're regulatory. Maybe instead of turning on effector T cells, they turn on Tregs. And so that led to a very interesting collaboration over the last several years. This is a map of where they occur. You can see they're in CH1. They're in CH2. They're not in CH3 of human and mouse IgG and IgG1 and IgG2. And some are in light chain. So they are spread out through the molecule. Some people believe they may play a role in even regulating an anti-idiotype response. We think they play a more general role. And this was published in Blood actually about 2.5 years ago. Wow. There's nothing unusual about them. These are human and mouse Tregs in terms of the structure. They all clearly have MHC class 2 binding motifs. They have anchoring residues. But there's nothing. I wish there was something that said this is going to be a Tregitope, is what they call it, and as opposed to an effector cell, but they look like regular epitopes. And this is what we think is going on with these cells, and I'll show you data that they actually have an effect. If this was our fusion protein, the Tregitopes could be here in FC. Actually, they wouldn't be in CH3, they'd be here. This might be a GAD or a MOG or whatever we've put on our fusion protein. This is presented by a B cell or a dendritic cell, and here's the effector cell binding. Here's a Treg that might be binding in close proximity, and that this Treg interaction leads to production of regulatory cytokines to shut down this effector cell. We don't know whether direct contact is necessary, and those studies are going on in a number of collaborators' labs. So this is what the hypothesis for what they think is happening. With Annie, we've set up collaborations with a number of different labs, which I think helps, in my mind, to validate this whole hypothesis. Federico Mengosi and Kathy High work at CHOP at Penn. They're interested in AAV gene therapy, where the immune response to the certain AAV serotypes is really critical, and there's some beautiful data that's going to be coming out on that. Nada Najafian and Mo Sayegh and Sami Akori and their colleagues at Harvard have done experiments using both EAE models and transplant models that, again, with these regulatory epitopes have shown suppression of the immune response. And Yansu in my lab and others, when I was at the University of Maryland, have shown additional studies. So just so I don't go over, I'm going to skip the data slides and simply show you that we can suppress the immune response to OVA by giving these regitopes, and we can actually suppress it in a TCR transgenic mouse, which I thought was pretty powerful. I'll just read you the summary of that. That we find a decreased amount of gamma interferon. We can actually see an increase in FOXP3 positive cells, and we seem to need natural Tregs for this to occur. So you need to probably recruit natural Tregs. There probably are adaptive Tregs as well. What about an autoimmune disease. And a number of experiments have been done over the years in my lab by Xin Li and others. And basically, if you treat animals at early in NOD females, you can prevent or reduce the incidence of diabetes. You delay the onset and reduce the incidence. Now, anyone who works in type 1 diabetes and NOD mice knows that you could treat animals over here early on where they may have peri-insulitis, but no, they have plenty of islets and good function. You can prevent diabetes by, well, there's probably hundreds of papers on how to prevent diabetes here. The more critical thing is relevant to the last talk, which is new onset diabetes. That's really, to me, where we need to go. Although we did some work with islet transplants, I think new onset is really where we need to go to be able to diagnose patients and start to treat them with an effective drug. So this is what we did. When non-females reached a blood glucose of 250 deciliters, milligrams per deciliter, we injected them with either PBS, in this case an IFA, because we wanted to create a depot, or a series of regitopes, in this case mouse T regitopes, or we left them alone. Now, we then followed glucose actually biweekly, and we simply looked for when these animals became diabetic. And this is a summary of the results. This is what happens if you take animals and as soon as they show a level of 250, do they continue on to become diabetic? And obviously within 2 to 3 weeks, they're actually, within a few days sometimes, they go up to 500 and we have to sacrifice them, which is why there are no more dots up here. On the other hand, if you just give them PBS, they continue on and 80% become diabetic in our colony. But if you give them the T regitopes, they actually level off at about 30 to 40%, and this is just one of 3 experiments with the same results. So to me, this is very encouraging in the sense that these regulatory epitopes have efficacy in diabetes. Seems to be some results working in terms of affecting CD8s in antiviral immunity and under certain controlled circumstances, and can work in a transplant model. And what we're working on now, or what the collaborators are working on now, is how to deliver these in the best way, because clearly we're not going to be giving incomplete Freund's adjuvants to patients. So that just is what I showed you before, what we think is the mechanism, and I should give credit that a lot of work has been done by other groups that make me feel more confident about this. In terms of what some people would say, did I drink a whole glass of Kool-Aid about this? I once made up a slide, wasted a lot of time with PowerPoint to show that it's only half full. I only drank half a glass of Kool-Aid. I still have lingering doubts about how far we can go with this, but I'm very encouraged in terms of the results we have so far. So when we think about antigens, whether it's autoantigens involved in autoimmunity or biotherapeutics, realize that we can map epitopes, but there also could be regitopes. There could be regulatory epitopes within proteins, not just immunoglobulins, but in other proteins as well. And if we can identify these, we should be able to modulate immunogenicity. And in addition, of course, you have to consider the biological properties, which is what I talked about first in terms of Factor VIII. And I'd like to cite the late Eli Surkars and work that was done as before some of the people in this room probably were born, not you, Noel. But what Barbara Arneo did in his lab is they took lysozyme and they cleaved off a C-terminal peptide, and it all of a sudden, from being a non-immunogenic protein, became immunogenic. And they suggested it was a suppressor epitope because at that time we referred to these as, suppressor cells. So I think we may be able to take proteins and manipulate them in ways in which we can make them either more tolerogenic by adding regitopes, or making even better vaccines by eliminating these regitopes, if we could figure out exactly what these epitopes are. So this is what we'd like to be able to do, is to make fusion proteins with regitopes, which actually look a lot like the immunoglobulin structures we started out working with. And again, I've already said this, that by using these, we may be able to develop new therapies and make recombinant proteins. Here's what we might envisage for Factor VIII. We can take the B domain out of Factor VIII, it still works, and we might be able to make a less immunogenic Factor VIII that would be useful. And you wouldn't have to do gene therapy. So to summarize, I've told you about factor VIII and thrombin, and the biological properties is important. I gave you some evidence about the IgG scaffold and that long-term interactions are important. And I've finished by saying that regitopes may be something we should focus on. It's a new term, not totally accepted yet, but I hope people will start to look at these and more people. If anyone is interested in this, it's very easy to set up collaboration with the DeGroot Group. And in fact, a PPG application is pending right now with all of these workers on it to apply it in a number of different disease models. So I'll stop right there, pretty close to on time. These are the people who did the work. I tried to refer to them as I was going along. Again, we had support from the NIH and a number of wonderful foundations. These are the people at Irvine and at Epivax, and I'll stop there and take questions. So thank you for staying this long.
In the last experiment that you show us with this model, what antigen was, what the antigen specificity was?
Actually, that's an excellent point. All we did is inject the regitopes in this vehicle to create a depot. We didn't actually link an antigen. We have done experiments where we've mixed regitopes with either pre-proinsulin or GAD, but in this case, we just gave them the regitopes. The reason being that we have a depot, we know that antigens are being released from the islets, and so that we figured there would be antigens present there. But it raises an important point. I personally don't think that you can be giving a peptide, whatever peptide you want to have that you think is a good immunosuppressive drug, alone, systemically. You ought to couple it to a target antigen, which is why I did that little cartoon at the end with the regitopes and a target peptide. I think I put everybody to sleep. Oh, there is a question in the back.
If you transduce B cells with your construct and then kill them and then inject them, could the apoptotic debris create the same effect?
That's a great question, and when all the work saying that apoptotic cells were very tolerogenic came out, Janssen did that very experiment, and what we found was that they were more immunogenic than tolerogenic. We were very disappointed in that one. And irradiated cells, well, irradiated cells normally die by apoptosis, but there may be some other subtle effects in it. But that just didn't work. Good idea. It's sort of like making fusion proteins that are going to be secreted by the host. Good idea, because that's the way it works. That's why you do research.
So how long these cells live inside the mouse, and what is the tissue distribution?
The B cells that are transduced,
Right.
we've gone out over 6 months. I think in Rachel's lab, they've gone even longer than that. You can find message in 10 months, and it's a pretty long time in the mouse's life. It doesn't mean that those B cells have that, but you can certainly see that a very long time. I'm not sure you even, well, when we actually, when Mary Litzinger was graduate student, she tried to trace these cells, and we could see them actually proliferating in vivo for several weeks, and then by 30 days, we've lost about half of them. By 60 days, we could barely find them. But again, the message is there, and we don't know what's happening in terms of turning on the Tregs. But the B cells with the constructs can last for a while, but the construct can certainly be protected. Maybe another cell for that. And the effect, by the way, with the B cells does last as long as 9 months. Like a single injection of B cells. Let's head to the traffic.
Okay, well, we have come to the end of another Autoimmunity Day. I want to thank the steadfast members of the audience for your participation. There are a number of people who made this possible. I think Mrs. Ladd may still be here. There she is, and she certainly has been the financial backer of this throughout the throughout its time. My secretary, Kathy, is in back and has done a wonderful job in keeping things together. My wife is in back and has kept me together. And I want to thank the two chairmen who kept us all on time. We have actually finished within a minute of the appropriate time, which again I think sets a precedent for other meetings. And to hope that we'll see you all back next year for another very successful Autoimmunity Day. So thank you all for coming.